First of all, thank you very much to the organizers for inviting me. And today I'm gonna share with you some results we have gathered looking at chromatin conformation temporally resolved during a circadian cycle. So as you might know, circadian biological rhythms are essential for organisms to anticipate and adapt to changes in the environment that are imposed by Earth rotation, so that effectively most physiology has a circadian component to it, from behavior as feeding and fasting and sleeping cycles to liver metabolism, including glycogen production, immune system activation, hormone production, and of interest in my lab, gene transcription. So every cell in the body has a self-sustained molecular clock, and this works as follows. These two transcription factors, clock and BMAL, will bind E-boxes in the genome and activate the transcription of several genes, including period and cryptochrome. And these two genes are transcribed, translated, and then the proteins go back into the nucleus and block the action of clock and BMAL, thus inhibiting their own transcription. On the other side, clock and BMAL also activate reverb genes, which are transcriptional repressors that will directly bind to ROR boxes in the genome and inhibit the transcription of BMAL and clock, thus preventing their own transcription as well. And these two negative feedback loops last around 24 hours and sustain the self-sustained oscillations in the cells. So apart from these core clock transcription factors, these TFs control the expression of several other output circadian genes in the liver, so that in total, the liver transcriptome, 10 to 20% of the liver transcriptome presents an oscillatory behavior, and you can see that here. This is a plot from an async experiment in which each row is a gene, and you can see how each gene presents a peak of transcription at different times of the day, with diurnal and nocturnal genes being expressed. And it is well known that not only transcription is rhythmic, but there are several epigenetic processes that can show also a rhythmic behavior. And this is true, for instance, for if we look at the binding of the core clock transcription factors in the genome, you can see that their chromatin occupancy is rhythmic, and it accumulates at different time points depending on the transcription factor. And this is also true, for instance, for the accumulation of certain histone modifications at regulatory elements. However, far less is known about how chromatin conformation at the different genomic scales behaves during a circadian cycle, and how this impacts circadian gene regulation. So we set out to answer this question, and to do so, we sampled liver tissue around the clock, 2 time points during the night, 2 time points during the day, and we performed chromosome conformation capture experiments, including Hi-C and promoter capture Hi-C, as well as RNA-seq. So for today's purpose, I'm just gonna tell you about the promoter capture Hi-C results. So this technique is, it starts from a Hi-C experiment that you might be well aware how it works. And then we take these Hi-C libraries and subject them to an enrichment step with the capture system. In this case, we designed probes to capture every gene promoter in the mouse genome, and like that you end up with a library that is enriched for promoter interactions between promoters or with promoters with other regions in the genome. This allows you to increase the resolution of the experiment by around 15-fold, depends on the enzyme you use. So by doing this, first of all, we asked, okay, so what is interacting with promoters? And we found what we expected, which is regions with regulatory element features. And of interest to us, we found this category of enhancers that have been described in the liver by producing eRNAs. And a set of these enhancers that are producing eRNAs show an oscillatory production of the eRNAs. So we ask, if you are an oscillatory enhancer, you tend to contact a circadian gene, and the answer is yes. So circadian enhancers are more than expected touching circadian promoters, meaning that oscillatory regulatory elements in the genome are coming together in the 3D space. Having observed this, we ask, okay, so at what time are these regulatory elements being transcribed? So what we could see is that if you're a diurnal circadian promoter, you tend to interact more with a diurnal circadian enhancer, and the same holds true for a nocturnal circadian promoter. You tend to interact more with a nocturnal circadian enhancer. We ask the same but for promoter-promoter interactions, as a subset of circadian genes are interacting with each other. And the effect here is even more striking. So if you are a circadian promoter, you tend to contact other diurnal circadian promoters, and the same holds true for nocturnal circadian promoters. You tend to interact with other nocturnal circadian promoters, right? So next, we wanted to explore the dynamic of the interactions around the clock. So we decided to count how many and at what time are circadian promoters with different acrophasers or transcriptional peaks establishing interactions. And what we could see is that for the different circadian promoters, the most interactions are established around the peak of transcription, and this is true for promoters peaking at CT0, 6, 12, or 18 hours of the day. So let me show you here an example. This is the interactome from the BMAL1 core clock gene promoter, and you can see here how this promoter is establishing these 3 contacts with enhancers at CT0. Then at CT6 and 12, there are no interactions being formed or detected, and at CT18 we recover these 2 interactions, and this nicely follows the expression of these genes that has its peak at CT0, then decreases at 6 and 12, and goes back up at CT18. So this is a very dynamic interactome with all the interactions engaging and disengaging during the day. However, this is not the case for all genes. For instance, here I'm showing you a circadian output gene, the GSK3A gene. And here you can see that this interaction profile is a hybrid one with stable interactions and dynamic contacts. And again, you can see that the higher number of interactions is established at the peak of the transcription of this particular gene, which is at CT12. So having observed these dynamic versus static interactions, we ask, okay, so which DNA binding motifs can we find in constant versus dynamic interaction? And we found several interesting candidates, and the top hits we found were these 3 up here. And interestingly, we saw NR5A2 also, that you have heard about in the previous talk, highly enriched in circadian interacting partners. And it is well known that this nuclear receptor is a key metabolic sensor in the liver, regulating lipid homeostasis, lipid synthesis, cholesterol homeostasis. So we verified through CHIP qPCR the binding of this protein at several of our loci, and we could see that even if the protein is not oscillating in quantity, the binding to these particular regions at least seems to be rhythmic, and we are now looking further into this phenomenon. So just to finalize, we also analyzed all the interaction profiles from the cord clock gene promoters and compared these to other circadian genes in the liver. And what we found is, as I show you for BMAL1, that cord clock gene promoters tend to establish very dynamic interactomes compared to other circadian genes in the liver, and they do tend to have slightly less contacts than other circadian genes in the liver. So let me show you here another example of that. So this is the landscape of interactions from the reverb alpha gene promoter, and you can see here how, again, we have dynamic interactions that are established around the transcriptional peak phase of the gene, which is CT6, and in contrast with this very saturated profile, which is fairly stable around the clock for this circadian output gene that peaks at CT0. So in summary, I didn't have time to talk to you about this, but you can have a look at the paper. So by doing Hi-C, we observed that TATs that contain circadian genes are constant during the day. However, they are switching chromatin compartments at the different time points. I did show you that circadian gene promoters can establish long-range interactions that can be dynamic or constant, and that these interactions are mainly connecting regulatory elements that are sharing transcriptional phase, either promoters or enhancers. And finally, I mentioned this interesting difference we are seeing regarding cord clock genes, which have fewer interactions that are really dynamic around the clock versus other circadian genes in the liver, which seem to have more stable, saturated contact profiles. So with that, I just want to thank my laboratory, the people involved in this project, Masami, Rodrigo, Abraham, and Luis, which are actively working, following up these results, and I'll take questions. Thank you very much.
Thank you very much, Mayra, for the nice presentation and very interesting theme to write of how you generate these interactions and how they are actually regulated also over time in the circadian rhythm. And so as before, I'm sure that there'll be questions coming on the Q&A. So can I please ask the audience to type your questions and so that I can read them to Mayra? But I'll get started with one which relates to your last slide where you very quickly discussed about the change of compartments, which I obviously also read in your preprint. And my question is a little bit the chicken and the egg, right? Do you think this is a consequence of the changes in the interactions? So this switch of compartments, or so can you have a little bit of at least temporal dissection of whether they could take place before and they could be facilitator of these interactions between promoters and enhancers? Or are they actually a consequence, or what are your thoughts on that? I mean, there's a lot of discussion in the field on that area, as you know.
Yes, so what we saw is that these changes in compartments correlates very well with chromatin modifications, also varying in these regions during the 24 hours, and transcription itself. However, with the resolution we have, it's hard to measure what happens first, if it's the chromatin, first the chromatin changes and then the interactions, or the other way around. Maybe to answer that, we could perform a higher resolution experiment focusing in a particular loci and measure these parameters 3 hours. Yeah, however, the compartments themselves from the data arise from the interactions, right? So it's really driven by the interactions. I don't know if there is a chromatin setup first. Yeah,
that facilitates those. Okay, thank you, Mayra. And yes, the questions have started to come, so I will ask. The first one is by Nicola. who's saying, nice talk and beautiful model. Do you have an idea of what are the features of the anchor regions that change over time to drive the changes in contacts? And he asks, is that transcription factor binding or chromatin marks? I think you discussed already part of it, but might be good to see whether you think, whether there's any features.
Yes, so we did find, as I showed, some particular TFs enriched at dynamic versus constant interactions during the day. So there seems to be a different protein composition, let's say, between the two, with some factors shared and some that are unique to each one. We didn't look into chromatin features, but that would be very interesting to have a look if regarding the epigenetic profile of those particular regions, if there are differences in chromatin conformation, even accessibility, or certain histone modifications. But in terms of transcription factors, yes, there are some shared ones and some that appeared only at dynamic or only at constant interactions.
Yeah, thank you very much. And there's a follow-up by Nicola, which I think I'll say because I think that follows a bit your answer. And what he says is, well, could these features be manipulated to suppress periodicity on the transcriptional response?
That would be a really nice experiment that we want to do. So start to manipulate the system and see if you have, let's say, circadian loopers that are more important than others, and establish the rhythmicity in the contacts.
Probably not so easy because these are really liver from the mouse, right? These are in vivo experiments, and that takes time to do. Thank you, Mayra. So next question is by Paul Delgado Holguin. Very interesting research, Mayra. Congratulations. For cycling enhancers, do you know if they are constitutively accessible or are activated, inactivated in cycles?
That's a very good question. This was not actually measured. So what is reported is that the transcription from them is dynamic. It has a rhythmicity to it, but I'm not sure if you look at accessibility, what would happen. It is known that circadian promoters gain and lose accessibility during the day in many instances. So I would expect this could be the same for enhancers.
And next question, Fernando Lopez Casillas. Do these oscillations depend on light-darkness cycles?
Yes, absolutely. So I didn't explain that, but so the light goes through the retina and synchronizes the central clock in our body, chiasmatic nucleus, and those neurons then signal the peripheral clocks in the body to synchronize them. So the synchronization of the clocks is dependent on light cycles. However, these clocks are already self-sustained in every cell in our body. So if you have a cell in culture, it's oscillating in transcription. So you don't need the input of the light anymore. These oscillations happen in all the body.
And so next question by Aidan Martens. Fascinating talk, thank you. What happens if you vary the periodicity/length of the light-dark cycle for the animals? Do the enhancer promoter dynamics change immediately?
This hasn't been measured. So now we want to start altering parameters that modify the oscillatory behavior in mice. One of those conditions would be what you are suggesting. Another one that we're going to start is modifying the diet and the time when food is consumed. So yes, those are very exciting questions.